Hello, my name is Myungjin Lee. I'm a bioinformatics scientist at Vaccine Research Center, NIH. Today I'm going to talk about GLYCO-2.0, which is a web-based server to quantify glycan shielding of glycosylated proteins with improved data processing and computational speed. So I'm going to talk about why I developed GLYCO program, and I'm going to move to the algorithm of GLYCO program, and then I'm going to talk about the applications, how GLYCO can be utilized in many other projects or research. First of all, why GLYCO? So in Vaccine Research Center, our lab main focus on the HIV/AIDS virus vaccine design. So this is the structure of HIV-1 enveloped trimer. And this trimer has GP120 that is colored in gray here. And then the bottom part is GP41 that is colored in yellow. And this protein structure is highly or densely covered with glycans that is colored in green here. And when I first saw this structure, I got a question that what is the relationship between the density of the glycan versus antigenic or immunogenic properties of the virus? And for lots of diverse glycoproteins, from the left, HIV-1 enveloped trimer, influenza virus, RSVF, Lassa GPC, SARS-CoV-2, or Zika virus, there are many different types of viruses that have glycans. And if you look at the structure, each glycoprotein has different density of glycan coverage. By comparing the SARS-CoV-2 versus Zika virus, for example, it's really easy to see which one is highly glycan covered or which one is low in glycan density. However, what is the glycan coverage comparison between Lassa GPC versus influenza virus? It is difficult to find out which one is higher in glycan density or which region in single structure is highly glycosylated compared to the other region. So I found that it would be really useful if I develop a program that can actually quantify the glycan density so that we can see how dense glycans covered each protein surface residues. And I searched some of the literatures, but I could not find any single work to computationally quantify the glycan densities. So I decided to develop an in silico program that is GLYCO. And GLYCO is from glycan coverage. So as the name says, GLYCO program is to develop to quantify glycan coverage of glycoproteins. So the goal of the study is to develop an in silico tool to quantify glycan coverage of glycoproteins. So now the algorithm. So what GLYCO program does is to take the input file as a glycoprotein. So this blue one is the protein surface, and this green one is glycan. So actually, a single input PDB file is glycosylated protein. And once the program takes the input file, what it does is take the protein surface residue. In this case, I colored in red here, so this is my protein surface residue of interest. And on each protein surface residue, what it does is to define a certain cutoff and then check the glycan atoms within the cutoff. And then GLYCO program generates each vector from the protein residue surface to each of the glycan atom and check if this glycan atom is actually giving the shielding effect to the surface atom. So it generates a vector in between the surface protein atom and the glycan atom, and then check if any of the protein coordinates is along the line of the vector, and if it is blocking this vector line, then the associated glycan atom will be excluded in the calculation. However, the one that does not have any protein in between will be included in the calculation. And the algorithm will collect just for the green one that actually gives the shielding effect to the protein surface and count the number of the glycan atoms. Then that would be our definition of glycan coverage. So the previous version that was published, which is GLYCO 1.0, adopts this kind of method for the key algorithm. So what it does is to generate the vector with a numerical parameterization, meaning that it generates the multiple number of dots instead of a line, and generates a cubic box for each dot, and this entire series of cubic box is my vector line, and check if any of protein coordinate is inside of the cubic box. So this one, this vector line has some of the protein atoms within the cubic box, so it'll be excluded, but this glycan will be included in the calculation. So GLYCO 1.0 is available at GitHub. So this is the link of the GitHub, and it was published in Bioinformatics this year. So if anyone wants to run GLYCO program, then you can freely download the script and then run your glycosylated protein. However, we found that it would be much easier if we develop a web server of GLYCO program so that anyone who are not really familiar with coding or programming can also use this program. And also, running multiple PDB files such as MD trajectory, the multiple frames from the molecular dynamics simulations, it has normally several hundreds of PDB files, and we found that there's a room to improve the computational time of GLYCO 1.0 so that we can better deal with multiple frames with hundreds of different PDB files. So in GLYCO 2.0, what we improved is that the first one is develop a web server, and the second one is improving the algorithm so that we can have efficiency in computation. So in the algorithm parts, instead of having multiple cubic box along the line of the vector in 1.0, we replaced the cubic box to the cylinder so that it can have a rigorous solution instead of having numerical parameterization. That's the first thing. And the second one is the web server development. So now GLYCO 2.0 web server is available online, and this is the link of the web server. And Matteo, who is another first author, did most of the work of server development. So if anyone wants to run GLYCO without doing all the code or programming, then you can simply go to this web server and then click the tab run GLYCO 2.0 and input your glycosylated, the glycoprotein. So now I finished all the algorithm and then the idea of the GLYCO program. And what is the output of the GLYCO? So the left structure is my glycoprotein, and if you run GLYCO with glycosylated structure, what you can see is on the right figure, glycan densities are all mapped on the protein surface. So by looking at this figure, you can easily see that which part is highly glycosylated, that you can see on the left and rightmost part, compared to the other, which is middle or bottom part of the structure. And GLYCO does not only take a single PDB, but it also takes multiple frames, multiple PDBs from MD simulations. So this is the 300 nanosecond of MD simulation. It has an average function, so that the right side you can see that it is the glycan densities are all averaged so that the gradation of the color is a bit different from the single PDB, but that it can better capture the glycan mobility and flexibility of the structure. And now I'm going to go to the application parts. So what we can do with glycoprotein. So first of all, which is a fundamental function of glyco, which is glycan coverage calculation. So this is the glycoprotein series that I showed in the previous slide. So if we run glyco, as you see at the bottom, you can easily see that which glycoprotein is highly glycosylated compared to the other one. And also in the single structure, specifically which part is dense in glycan compared to the other parts. For example, HA, influenza HA is highly glycosylated, some part of the stem, but then the side parts are kind of sparse in glycan. But Zika virus, there are only 2 glycans are here, so the mostly are sparse in glycan except this 2 part. So we can easily see that the glycan density per each region, and also comparison between 2 different structures. This is another example of glyco application. We calculate the glycan density for the influenza virus subtype H3N2 from 1968 to 2020, and we found that glycan density on the head part of the influenza virus is being accumulated as time goes on, so that you can see the clear plots of increasement of the glycan since pandemic 68. And also by having glyco results, you can see that specifically which region is highly glycosylated compared to the other parts. So in HIV-1 trimer, it has names on each region. The top part is called V1, V2 region, and the left and rightmost part is called glycan V3, and then the side part is called silent phase. So these 3 regions are relatively high in glycan density compared to the other parts. And also the top view is showing that the top part of the structure is mostly evenly covered by glycans. Another application of glyco is to find out the glycan hole location. So on the left figure is my HIV-1 wild type structure that you can see that the top part is mostly glycosylated. However, if we take out 3 glycans, actually there are 3 glycans at N160 in BG505 wild type structure. Once we remove these 3 glycans, we were able to find out that glycan hole was generated. So glyco has an ability to find out the glycan hole by calculating the glycan density, and glycan hole is a really critical place in vaccine design, because normally antibodies try to target the glycan depleted area so that it neutralizes the antigen. So it is a really critical observation of glycoprotein. This paper has been published in 2021 of SARS-CoV-2 structure. So what I did was to calculate the glycan density of the SARS-CoV-2 structure throughout. And there's a place that is called NTD supersites, where the antibodies are targeting mainly. And we proved that glycan density on the NTD supersites is relatively low compared to the other parts, so that it is a vulnerable target of antibody binding. And next category of the application is to correlate with antigenic properties. So recently we found that the antibody binds with specific distance to the antigen. So for this complex showed that antibody VRC26 and PGT145 has substantial distance when it binds to the antigen. And we actually measured the distance for all kinds of HIV-1 antibody and colored the distance from white to red, so that if the distance is really low, then it'll be in gray or white. If the distance is really high, it'll be in red. So once we found all this distance distribution, I got a question that what is the relationship between the distance versus glycan density? So GLYCO has a function to calculate the glycan density just for the specific epitope. So for example, epitope called V1, V2 region is colored in cyan here. We calculate the glycan density just for the V1, V2 region. And next one, which is glycan V3 epitope, we also calculate the glycan density just for this region as well. And I repeated all the epitope region, and then try to find out the correlation between the glycan coverage and distance that I calculated in the previous slide. And then we were able to find out the high correlation between the glycan density and antibody antigen binding distance, meaning that if the antibody is approaching to the high glycan density region, then it tends to have longer distance. Rather, if the antibody approaches to the low glycan density, then it rather has lower, shorter binding distance. The last application of GLYCO is defining a new metric. So this is the HIV-1 structure with glycans everywhere, but except the base parts. So we found that we have to cover this base region because it is exposed and it is realistically not, it does not really exist in real life, because this is the membrane connecting region. So in order to cover this part, we wanted to glycosylate this base residues. But before we do that, we had to define the base residues. There is no concept of base residues at this point. So what we did is to use GLYCO program to calculate the glycan coverage of the HIV-1 structure and color them binary versus green versus white. And if there's no glycan coverage, then we define that residues as our base residues. And then from that point, we can continue the all kinds of research that conservation study, and we found that the base residues are mostly highly conserved, except 3 residues that is colored in purple here. So the whole idea of doing this is to define a new metric. So GLYCO has an ability or utilization to define a new metric that has not been reported or achieved before. So the summary, in silico glycan analysis program, GLYCO was developed to quantify glycan density, and GLYCO is the only one method available in public to calculate glycan coverage. And GLYCO can be utilized in diverse work to understand the distribution of glycan and its coverage. And I'm going to show how to run GLYCO 2.0 web server by running my tutorial. I'm going to show how to run GLYCO 2 web server. First of all, open a browser and type glyco2.niaid.nih.gov. Then you'll find the GLYCO 2 web server. And if you scroll down, you'll see there are a lot of parameters that you have to select or enter. But there's only one input file that you have to prepare to GLYCO 2, which is your glycosylated protein PDB file. So I'm going to use -5 glycosylated influenza HA. So in order to enter your input file, click choose files and then select your input and enter. If you go down, you have to select your module type. So there are two different modules. First one is all protein atoms that calculates the glycan coverage for the entire surface. And second is subset that calculates the glycan coverage just for the specific regions that users define. So I'm going to use all protein atoms because I want to calculate the entire surface. And minimum RSA is the cutoff to define the surface protein residues. Normally 30 is the reasonable value to define the surface protein residues, so I'm going to use it as it is. And then you have to enter your glycan residue name. So in my PDB file, I have 3 different glycan names, which is BGL, BMA, and AMA. So I'm going to enter BGL, comma, space, BMA, comma, space, and AMA. And below that, you have to decide your distance cutoff to define the glycan shielding. I'm going to use 23 here. And cylinder radius, I think you can keep 1.4 as the default as it is. And then name your project, which is HA Glyco, and type your email address so that you can get the notification when the job is finished or job is received. And then click submit. Now the job is submitted, and soon you'll get an email saying that the job is successfully submitted, which I just received. It will take some time to calculate the glycan coverage. So in the meanwhile, I can show the example output that was already done, which is this one. So this is the output folder, and in the output folder, you'll see 4 different files. Out of these files, what you want to focus on is the first one, bfactor.pdb. So I'm going to open this one in PyMol. Let me first close the other one and then open it here. So what you want to do is to type spectrum space B, comma, white, green, black, and enter. In order to visualize, I'm going to extract glycan by select organic and then extract it as an object. Now you can see that the protein residues are colored from white to green to black based on the glycan coverage. And in order to visualize better, I'm going to change the representation to surface. Then you can clearly see that which part is highly glycosylated and which part is sparse in glycan. Yeah, this is the web server tutorial, and I'm gonna conclude my talk by acknowledgements people from the structural biology section in NIH, and also structural bioinformatics core section, and also collaborators with NIH Vaccine Research Center. And this is the group photo from our lab, and thank you for listening to my talk.